Hello everyone, I am Sanjota Ji. In this session, I will be discussing introduction and scope of microbiology. This comprises historical perspectives, important contributions, scope and branches of microbiology. As we all know, the microbiology is the study of microorganisms which are microscopic in size. This term was first introduced by French chemist Louis Pasteur. His period was 1822 to 1895. He demonstrated fermentation process and explained that this process was caused by the growth of bacteria and yeast. Later, the term microbe was used by Sidilot in 1878, and later now it is called as microorganisms. How the microorganisms were observed and who observed for the first time? The answer is Antonie van Leeuwenhoek. He was the first person who constructed the microscope which was designed by him, and he observed the different shapes of bacteria, and hence he is called as father of bacteriology. Let me explain about the microbiology. Microbes are ubiquitous in nature. That is, they are present everywhere. They can be viewed only with microscope. We can't observe the microbes by using our naked eye. The size of the bacteria range from 0.2 to 1.5 micrometers in diameter and 3 to 5 micrometers in length. They are listed from smallest organisms such as prions, viruses and bacteria, and to the largest microbes like fungi and parasites. These microbes, which are present everywhere, those are of two types. One is beneficial microbes and another one is harmful microbes. Beneficial microbes are the organisms which are used to produce various useful products like antibiotics, pharmaceuticals, etc. But harmful microorganisms causes various diseases. As we all know, now pandemic is there, coronavirus. So it is caused by virus. Not only virus, the bacteria, fungi, many of the microbes causes various dangerous diseases to human being. Hence, they are categorized under harmful organisms. The bacteria is the microbe which is used by different scientists. Let me explain the history of microbiology. In ancient time, the actual discovery of microbes was carried out before many centuries. The Mahavira's teachings was used in early 6th century, that is before Christian era. The Jainism reported the existence of invisible microbiological world that was depend on Mahavira's teachings. In the age of Islamic civilization, an Iranian scientist invented the presence of microorganisms such as Avicenna in his book The Canon of Medicine. Avicenna hypothesized the existence of microorganisms. He is regarded as one of the most significant physicians, astronomers, thinkers, and writers of Islamic Golden Age. He is called as a father of early modern medicine. In 1665, Robert Hooke observed fruiting bodies of moulds for the first time. He used microscope for the observation. Later, in 1676, Antonie van Leeuwenhoek, as I have already explained you, that he viewed the bacteria and some other microorganisms through a single lens microscope which was designed by him. Hence, he is known as father of microbiology. In earlier days, many of the biologists have speculated that there was a existence of living things from non-living things. That is called as spontaneous generation. So this spontaneous generation was disproved by Louis Pasteur by explaining the living things comes from living things itself, and he explained that by conducting the experiment called as swan neck flask in 1859. Later, in 1859 itself, the Charles Darwin who published The Origin of Species. We have studied earlier history or ancient history. Now we will discuss modern history or the age of biotechnology. In 19th century, Louis Pasteur and Robert Koch were considered as fathers of modern microbiology. In 1919, Carl Erecke is called as father of biotechnology, who has started first time the biotechnological process, who was explained. In 1928, Fred Griffith was conducted the experiment called as transformation process. It was explained by him he used Streptococcus pneumoniae organism to explain the transformation process. In 1933, electron microscope was invented. In 1937, living organisms were first divided into prokaryotes and eukaryotes. Beadle and Totem proposed one gene, one enzyme hypothesis. They explained that gene is responsible for the expression of particular protein, and they explained that in 1940s. Later in 1941, Oswald Avery proved that DNA is the genetic material. Actually, this was explained by Griffith, who conducted transformation experiments, but later Avery, he explained the DNA is the genetic material and he proved that. In 1953, Watson and Crick found that DNA is a double helix model, and they explained the structure of DNA in detail. In 1960s, Jacob and Monod discovered operon theory and explained the controlling of enzyme expression, and they explained lac operon theory and tripoperon theory, etc. In 1977, Fred Sanger invented DNA sequencing techniques. DNA sequencing, the meaning of this is we can study the bases present in the DNA. Till now, we have studied about history of microbiology. Now we will learn important contributions of microbiology. In the period 1632 to 1723, Antonie van Leeuwenhoek, he was the first person to observe microscopic organisms. This I have already explained. During 1749 to 1823, Edward Jenner has produced vaccines against smallpox. Later, Louis Pasteur observed microbial fermentation and he conducted many experiments. Seminal discoveries in vaccination was studied by Louis Pasteur and he also explained germ theory of diseases. Later, during 1827 to 1912, Joseph Lister, he introduced the sterilization techniques. Sterilization is a process of removal of all types of microbes from the area or from the surface or from the media, etc. Robert Koch, he was the German scientist who received Nobel Prize for discovering medicine for tuberculosis during 1843 to 1910. He also identified causative agents of tuberculosis, cholera, and anthrax. During 1850 to 1934, Hesse observed the agar substance. It is the substance which is produced from seaweed and it is used for the solidification of media. During 1851 to 1931, Martinus Beijering first observed the viral and bacterial nitrogen fixation and sulfate reduction in bacteria and virus. During 1853 to 1938, Hans Christian Gram, he was the Danish scientist who explained the Gram staining technique. This is a staining technique which is called as differential staining and is used to differentiate gram-positive bacteria and gram-negative bacteria. During 1857 to 1940, Julius Wagner Jauregou received Nobel Prize for discovering medicine for neurosyphilis, which was treated by inducing fever with malarial parasites. During 1885 to 1948, Marjorie Stephenson is the pioneer of bacterial metabolism and he explained the metabolism in bacteria. Kiyoshi Shiga, a Japanese scientist, identified the dysentery which was caused by bacteria during 1871 to 1957. Later, during 1854 to 1917, Emil Adolf von Behring received Nobel Prize for discovering medicine for diphtheria and anthrax. Later, Sir Ronald Ross discovered medicine for malaria, which was transmitted by mosquitoes during 1857 to 1932, and he received Nobel Prize for his work. Let's talk about a renowned antibiotic, penicillin. We all know that it was discovered by Alexander Fleming, but we did not know the other two scientists also discovered the same penicillin antibiotic, who are Ernst Boris Chain and Howard Walter Florey. During 1881 to 1979, during this time, the penicillin was discovered by all these three scientists. Later, during 1888 to 1973, Selman Abraham Waxman who discovered streptomycin and all other antibiotics. In 1980s, genetic manipulation and polymerase chain reaction was developed by Kary-Mullis. Later in 1990s, genetically modified microorganisms or genetically engineered microorganisms were studied, and prions or specific protein pathogens were developed, and the well-known Dolly the sheep developed by Wilmot. We have learned history and the various contributions of microbiology. Now we will study scope of microbiology or applications of microbiology. Microbiology is one of the major complex biological area, and it deals with various distinct biological fields. Natural history of microorganisms involves interaction between microbe, human, and environment. The nature microbes perform geochemical cycles like cycling of nitrogen, carbon, and phosphorus. Let's talk about the applications of microbiology in various fields. First, we will study bioremediation. What is this bioremediation? It is the process where utilization of microbes which are present in nature, or you can add microbes to the area where the more pollutant is present. The microbes which you have added are the microbes which are present in the nature. They will cleave that pollutant, and it will decreases the pollution in the environment. Next one is biopesticides. What are these biopesticides? These are the types of pesticides which were obtained from the natural sources such as animals, plants, bacteria, and certain minerals. Later, in medical field, what is the application of these microbes? Various microbes cause diseases like variola virus, which causes smallpox. Not only this, many of the microbes like bacteria or fungi, they cause different diseases in human beings, also in animals and plants. It is about harmful effect of microbes and the study of the diseases which are caused by which organism, that all can be performed in medical field, and along with this the microbes also perform activities such as controlling many diseases by antibiotic production, and other novel drugs are produced from the microbes, and they are used for the curing of very dangerous diseases. Let's talk about the curing of genetic diseases by gene therapy. Gene therapy is the technique of introducing the therapeutic delivery of nucleic acids into the patient's cell to treat the various genetic diseases. Next, we can study food industry. What is the application of microbiology in food industry? If we talk about food industry, we can see only harmless effect of microbes. But apart from harmless effect or beneficial effect, the microbes can cause various dangerous effect on food. What is that? We'll study now. So microbes play a major role in producing various foods like yogurt, cheese, wine, bread, SCP, single cell oil, single cell protein, pickles, soya sauce, vinegar, etc. etc. So many of the like bakery food or any of the beverages can be produced from the fermentation process by using microbes. This is beneficial effect. But what is the harmful effect in food industry? The microbes cause food spoilage or food poisoning. Okay, the example is Staphylococcus aureus, some of the mycobacteria. Those cause food spoilage. They release toxin to the food and they cause diarrhea in human being. Next is biotechnology. How the microbiology has applications in biotechnological field. So here we can produce many of the solvents like alcohols, acetones, organic acids, and also enzymes. Many new drugs also can be produced from the microbes, but using biotechnological technique called as genetic engineering technique. So rDNA technology is also called as genetic engineering technology, and it is used to synthesize many of the therapeutic substances like human growth hormone, insulin production, and interferons also can be produced by using genetic engineering technique, which comes under biotechnology. Later, how the research takes place or what are the applications of microbiology in research field. Here we can produce millions of identical copies in very short period at low cost, can synthesize huge number of homogeneous experimental substances. In research, we can produce identical copies. For example, transgenic plants, if you want identical copies like clones, can be produced by using microbes, and in a short period you can produce those, and huge number of homogeneous experimental substances also can be produced by using microbes in research field. How microbes play a vital role in industry. The various microbes are used in commercial and industrial purposes. Agriculture microbiology. Here we can study transgenic plants and animals. We can produce transgenic plants and animals from the genetic engineering technique. We can apply genetic engineering technique and produce transgenic plants which can yield very soon, and we can get more yield within a short time, and many of the resistant plants you can produce like disease resistant, insect resistant, pest resistant against various environmental factors. Later, geochemical microbiology. Here we can study bioleaching. It is the process where recovery of minerals from low-grade ores. This also is done by using microbes, and role of microorganisms in geochemical cycle. That is nitrogen fixation, phosphorus, carbon fixation, that all can be done by microbes. Till now, we have learnt historical perspectives, important contributions, and scope of microbiology. Now, we will discuss what are the branches, how many branches of microbiology are there. First one is bacteriology. It is the study of bacteria. Next, mycology. It is the study of fungi. The study of protozoa is called as protozoology. Later, the study of algae, it is called as algology, parasitology, the study of parasites, immunology, the study of immune system, virology, the study of viruses, nematology, the study of nematodes, and microbial cytology is the study of details of microorganisms. Next comes microbial ecology. What is this microbial ecology? It is the relationship between microbes and environment. As we have already studied, that is geochemical cycles, that is nitrogen fixation. It is the actual study of microbial ecology, that is the relationship between the microbes and environment. What all the bioremediation, biopesticides, that all comes under this microbial ecology. The microbial genetics. Here we can study the microbial genes and organization and regulation of microbes. That is genes. How the microbial genes organized and regulated. That is expression, or we can call that as a molecular biology. Next one is cellular microbiology. Here we can study bridge between cell biology and microbiology. Cell biology deals with the various components of cells can be studied, but microbiology is the study of microbes. What is cellular microbiology? The study of cells, unicellular organisms, and the components can be studied by using this. Next one is microbial taxonomy. Here we can classify the microorganisms into different forms, and naming of those microorganisms can be studied by using microbial taxonomy. Nanomicrobiology. Nano level. If we study the microbes on nano level, then it is called as nanomicrobiology. Let's talk about applied microbiology. Which are the applied microbiologies? Medical microbiology. Here we are studying pathogenic microbes. What is the epidemiology caused by microbes, and immunology, how the immune system is produced against microbes, and human microbiota means what all the microbes which are present inside the human body, that also can be studied, which comes under medical microbiology. Next one is pharmaceutical microbiology. It is the study of microbial pharmaceutical products like antibiotic production or new drug production, that all can be carried out in pharmaceutical microbiology. Next one is industrial microbiology. Use of microbes in industrial process. As I have already discussed earlier, that the production of various food products and also beverages and many solvents, those from the microbes by using industrial process. Those that all can be studied by using industrial microbiology. Next one is phylogeny. Phylogeny is the genetic relationship between diverse microorganisms. That means if many microorganisms which are somewhat similar, but the inner DNA content or the expression or characters of those organisms are bit different. So what is the relationship between the different microorganisms can be studied by using phylogeny. Next branch is microbial biotechnology. What we will study in this area? Manipulation of microbes using genetic engineering techniques. at the genetic and molecular level. I mean, the production of genetically engineered microbes. If the microbes which are able to produce very less product, then by using genetic engineering technique, you can introduce any of the new gene and you can induce that organism to produce more amount of that product. That can be done by using microbial biotechnology. Next one is agriculture microbiology, study of relationship between the plants and microbes such as nitrogen fixation. Veterinary microbiology, study of the role of microbes in veterinary medicine. That means various microbes which cause the diseases to animals, that also can be studied, and the various medicine which is produced from the microbes that can be applied to the animals. So this both can be studied by using this veterinary microbiology. The another branch of microbiology is environmental microbiology. Here we are going to study the diversity and action of microbes in natural environment. The best example we can give is bioremediation. The microbes which are present in the environment can remove the pollutants which are present in the environment. So that can be studied by using environmental microbiology. Next one is water microbiology. Whatever the microbes which are present in the aquatic ecosystem, the study of those microorganisms is called as water microbiology. Next is aeromicrobiology. Microbes which are present in the air, that can be studied by using aeromicrobiology. Last one is biotechnology. We all know that it is concerned with the genetic engineering. We can use genetic engineering technique to produce genetically modified organisms or transgenic plants or transgenic animals which are more beneficial. These are the some references which I used to prepare this session. Thank you.